high-yield USMLE-style questions focused on cell trafficking, vesicular trafficking proteins, Golgi function, and lysosomal storage disorders, such as eye cell disease. Question 1. A patient presents with coarse facial features, gingival hyperplasia, corneal clouding, and restricted joint movement. Genetic testing reveals a mutation in the N-acetylglucosamanyl 1-phosphotransferase gene. Which of the following is most likely responsible for these symptoms? A. Defective lysosomal enzymes due to a failure of mannose-6-phosphate phosphorylation. B. Dysfunctional signal recognition particle, SRP. C. Failure of copy vesicles to transport proteins from the Golgi to the endosome. D. Disruption of clathrin-mediated endocytosis. E. Failure of covesicles to transport proteins from the ER to the Golgi. Answer: A. Defective lysosomal enzymes due to a failure of mannose-6-phosphate phosphorylation. Explanation: eye cell disease, mucolipidosis type 2, is an inherited lysosomal storage disorder caused by a defect in N-acetylglucosamanyl 1-phosphotransferase, which results in failure to phosphorylate mannose residues, mannose-6-phosphate, on glycoproteins in the Golgi. This prevents proper targeting of enzymes to the lysosomes, causing them to be secreted extracellularly, leading to the accumulation of undigested substrates in lysosomes, resulting in symptoms like coarse facial features, gingival hyperplasia, and corneal clouding. Question 2. Which of the following is the role of copy vesicles in eukaryotic cell trafficking? A. Transporting proteins from the Golgi to the ER. B. Transporting proteins from the ER to the Golgi. C. Mediating receptor-mediated endocytosis at the plasma membrane. D. Trafficking proteins from the cis-Golgi to the trans-Golgi. E. Facilitating retrograde transport from the Golgi to the ER. Answer: B. Transporting proteins from the ER to the Golgi. Explanation: copy vesicles are responsible for anterograde transport, moving proteins from the ER to the Golgi. This is essential for the proper flow of newly synthesized proteins that need further processing and sorting in the Golgi before being sent to their final destinations. Question 3. Which of the following cellular structures is responsible for adding mannose-6-phosphate to proteins, a critical modification for lysosomal targeting? A. Rough endoplasmic reticulum. B. Golgi apparatus. C. Lysosomes. D. Cytoplasm, nucleus. Answer: B. Golgi apparatus. The Golgi apparatus is responsible for post-translational modifications such as the addition of mannose-6-phosphate to proteins. This modification is crucial for targeting enzymes to the lysosomes. A defect in this process, as seen in eye cell disease, prevents proper lysosomal enzyme targeting and results in the accumulation of undigested material in lysosomes. Question 4. A patient with a history of chronic alcoholism presents with liver dysfunction. Which of the following processes is most likely impaired in the hepatocytes of this patient due to changes in smooth endoplasmic reticulum function? A. Lipid synthesis. B. Protein synthesis. C. Glucose 6-phosphatase activity. D. Detoxification of drugs and poisons. E. Glycogen storage. Answer: D. Detoxification of drugs and poisons. Explanation: the smooth endoplasmic reticulum is involved in detoxification processes. In chronic alcohol use is upregulated to handle the increased demand for detoxification of alcohol and other substances. Question 5. Which of the following best describes the function of clathrin-coated vesicles in cellular trafficking? A. They mediate receptor-mediated endocytosis and transport proteins from the trans-Golgi to the lysosomes. B. They transport proteins from the ER to the Golgi. C. They are involved in phagocytosis of foreign particles. D. They mediate the transport of proteins from the Golgi to the endoplasmic reticulum. E. They facilitate translocation of ribosomes in the cell. Answer A. They mediate receptor-mediated endocytosis and transport proteins from the trans-Golgi to the lysosomes. Explanation: clathrin-coated vesicles are involved in receptor-mediated endocytosis, e.g., internalizing LDL particles, and also help transport proteins from the trans-Golgi to the lysosomes. Defect in the signal recognition particle, SRP, is likely to cause which of the following? A. Accumulation of misfolded proteins in the Golgi. B. Impaired ribosomal targeting to the rough endoplasmic reticulum. C. Failure to remove introns from mRNA. D. Disruption of lysosomal enzyme targeting to the Golgi. E. Impairment of CoP vesicle function. Answer: impaired ribosomal targeting to the rough endoplasmic reticulum. Explanation: the signal recognition particles RP is a cytosolic protein complex that guides the ribosome mRNA complex to the rough endoplasmic reticulum for protein translation. If SRP is defective or N, which of the following is the role of early endosomes in cellular trafficking? A. To transport proteins from the Golgi to the plasma membrane. B. To degrade misfolded proteins in the lysosomes. C. To sort and direct materials either to the lysosomes for degradation or back to the plasma membrane. D. To package proteins for secretion outside the cell. E. To facilitate the synthesis of phospholipids. Answer: C. To sort and direct materials either to the lysosomes for degradation or back to the plasma membrane. Explanation: early endosomes are involved in sorting and directing internalized materials, either for degradation in the lysosomes or for recycling to the plasma membrane or Golgi. 8. A patient presents with a genetic disorder that results in defective lysosomal enzyme targeting due to a failure in mannose 6-phosphate phosphorylation in the Golgi apparatus. Which of the following is the most likely consequence? A. Defective secretory protein production. B. Accumulation of undigested cellular debris in lysosomes. C. Impaired endocytosis of extracellular materials. D. Decreased protein translation in the rough endoplasmic reticulum. E. Increased synthesis of glycoproteins in the Golgi. Answer: B. Accumulation of undigested cellular debris in lysosomes. The